Hey everyone, and welcome to tonight's Blue Rose webinar. I'm joined by a cat climbing up my chair, little mountaineer next to me. But anyway, so let's talk a little bit about a big shift happening in genomics. So the big shift that I'm referring to is a move from single reference genomes to pangenomes. Now, before I get into all of the details of that specifically, let me add, as always, that I am a human being. I haven't even completed a PhD yet, so I only have a couple of degrees. I'm not an expert in many of the things I speak about. I only know the basics, if even sometimes. And so if I do make mistakes, which I inevitably will, by all means correct me. I'm just relaying the little bit of information that I happen to know, and it is a little bit. So if anyone has a lot more to add to that, please do. I'm not claiming to be an expert. I am just relaying other experts' information. Um, but with that being said, so tonight's webinar is all about the specific experts whose information I'm relaying tonight is an example from one of my favorite journals, Nature. So what they did is they looked at sort of the way that reference genomes are assembled, and they looked at something called a pangenome. So just for a bit of nomenclature there. So a reference genome is the standard sort of DNA sequence we use like a map. Most clinical pipelines align a patient sequence reads to these reads to find the different variants and sort of differences. And when you look at the gene discovery channels, you always look at the reference genome to see how does that relate to variability. Now, most clinical pipelines align a patient's sequencing reads to find the differences in a very Eurocentric um, genome. So the Eurocentric bias is what it's called, and it's essentially referring to the fact that a lot of the people who were used in past historic sort of data collection methods were all from European descendants. And so um, the problem is that a reference genome is ultimately based on a very small number geographically, so it can't represent everything that exists in real populations. And this is especially prevalent in South Africa as well, where we have a lot of research going on. I did my master's in one example like this, where we look at my master's was specifically in African genomes, and there's a lot of research going on about sort of where they look at South American populations, and the specific Nature publication that I'll be referring to as my designated example tonight is one where they looked at different Chinese populations. So, yeah, like I said, a pangenome is the idea that instead of one map, essentially, if you think of a genome as a map, we build a map of maps. So it's a representation built from many individuals' entire genomes. So we can capture DNA sequences and variants that are missing from the current reference genomes. So the paper that I'm speaking about, the one that I'll reference from Nature, as always, I'll put a link to my little example, which is the good example of this exact case, which where they used the 1,000 Chinese pangenome project. So it's the 1KCP project. There's also a 1,000 genomes project, Africa Genome Project, and there's a bunch of these sort of 1,000 genome projects and 1,000 pangenome projects and that type of thing. But the specific one is one where they looked at over 1,000 high quality genomes that were assembled to reveal hundreds of millions of DNA letters that aren't in the main genome references, the human genome references. And many of them are either in or at least near medically relevant genes. So, in other words, genes that inform on drug prescriptions or genes that informed on uh, transplantation, immune response likelihood, and etc. That type of thing. So, in the clinic, most people hear genetic variant, they immediately think of a SNP, a single nucleotide polymorphism. So that's a one-letter DNA change, like an A becoming a G or a C becoming a T. And SNPs are incredibly important, right? They are kind of the bread and butter when it comes to genetic variation, but they are not the whole story. There's two major classes um, that often just don't go reported on, even though they're extremely valuable as well. So the one is something called structural variants, so SVs, structural variants, and essentially what these are uh, kind of they large changes like deletions and duplications and inversions and insertions. So, and when I say large, I mean we're talking about 50 base pairs or even larger at a time. And so you can kind of think of these if you use the analogy of letters in DNA code, then these would be like sentences or paragraphs or however you want to, so longer sections. And so deletions, missing part, a duplication, it's repeated. Well, put an asterisk next to repeated. I'll tell you why in a second. So it's the entire section that's repeated, duplicated, kind of like a copy paste, inverted. So remember DNA. That's why I used this, it was so emphasized so much the analogy of code or letters, because a lot of people, when you speak about the code of DNA, there's a lot of people that seem to think it's a literal code. These letters A, G, C, T, it's 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 shorthand for molecules. It's not literal computer coding. And I know that sounds ridiculous to specify that, but there are genuinely people who think it's literal letters. Yeah, so and so if you think about the actual molecular structure of DNA, and the fact that it's a double-stranded thing, an inversion it wouldn't really make sense if you just think about it in terms of here's the sequence and now it's the other way around. But that doesn't make sense, because how does it bind on the backside or on the phosphate group side chemically? That doesn't make sense. But then you remember there's two directions. There's the forward strand, and then there's the reverse strand. And the reverse strand is the one that can go onto the other side. And then you have a case where the letters are read the opposite way around. So it essentially inverts it like that. Um, or I like that for the that's not very useful for the people who are only listening, because there are a few viewers. Well, I suppose they're not called viewers, but there are a few listeners who don't have the video access part of this webinar. But yeah, so an inversion can happen in multiple different ways. And one way to make sense of the way that it chemically binds is either it's the reverse strand that's being flipped sort of over its backside, or it can be the 5 prime to 3 prime one that's sort of switched around on the horizontal axis. If you think about it in a 3D space, it's quite difficult to paint that picture. Or for me, it's quite difficult to paint that picture. Maybe someone else would have had a better vocabulary to describe that. So yeah, you can think of these structural variants as sort of the paragraphs that went missing or duplicated or flipped or copy pasted. And by the way, so an insertion is essentially, so let's say if a gene sentence got copied, and then instead of placing it directly after where it was already, is if it's kind of like pressing Control X, you're taking it out and you're inserting it way somewhere else in the genome. So that would be an insertion. So those are just examples of structural variants. Another big variant is one that we see in all sorts of neurological disorders, like Huntington's disease, and these are tandem repeats. So these are short DNA motifs that are repeated back to back, like CAG, 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 or GTC, GTC, GTC. So it's just the repetition. And it doesn't specifically necessarily have to be 3 letters, although those are more common, because just if you think about the mechanics of how something goes from DNA to RNA to eventually a protein, is it reads, the transcription happens 3 letters at a time. That's called a codon. So it codes 3 letters at a time to make an amino acid, which makes a polypeptide chain, which folds into a protein. That whole central dogma thing that we mention a lot of times in these webinars, where you go from DNA to RNA to protein. And so it's very often 3 letters, but it doesn't have to be. In fact, I think, and correct me if I'm wrong, but I think Huntington's disease is just the 2 letter repeat. But I could be wrong. So if I'm wrong, please correct me. But yeah, so that's a repeat or a tandem repeat. So clinically, classifications depend heavily on allele frequency, AF. So that's essentially just saying how common is this variant, and I specifically say variant instead of SNP or allele. So how common is this variant in the specific relevant population that you're looking at. Because uh, just to elaborate on that, what is the major allele or the common allele or the normal allele in one population may not be the normal allele in a different population. So this is it's very contextually important. What population are you looking at when you're looking at alleles? And it's the same if you go beyond. So this is all medical population genetics, but if you go take an example in the animal kingdom, I think when they talk about these different alleles and sort of survival benefits, if you think about the black pepper moths, if the moths, some are black, some are white, or the wings are like kind of brownish and speckled, and in the areas where it's say snowy or whatever, or the bark is white, then the ones with the light sort of bark colored wings are more likely to survive. And then in what happened, which was quite interesting in the during the industrial era, where there was a lot of soot on the trees and sort of stained them black, the ones with the black wings were more likely to survive and not get eaten by birds and stuff. So the allele frequency changed, and the common allele became the black wing allele. And so yeah, this is just one example in an animal case where the major allele, depending on the area, can change depending on what is more beneficial, depending on historical context, and etc. etc. There are so many different factors, environmental factors that actually ultimately influence why some alleles are, or the allele frequency can change from population to population, and geography is often a very, very important factor in this as well. So if your reference data set doesn't necessarily represent your patient or their ancestry at least, you can essentially misjudge how rare a variant really is, and this can absolutely distort interpretation. And so this project that I'll be referring to, the one that's published in the Nature publication, where they looked at a Chinese population sort of trying to tackle this problem directly. So the one of having different genomes or different variants that are not necessarily looked at in the common reference genomes. There are two. I speak about the common reference genomes a lot. So there are two very common ones. The one is called GH GHCH GR. There we go. GRCH38. Yeah, GRCH38. That's the common human genome reference. And then the other very common one is the CHM13. CHM13. Those are the two common. So it's difficult to remember all of these. Same with the genes with all of these numbers in their names. It's very difficult to remember sometimes. But um, yeah, so those are the two common genome references that are widely used as human reference genomes. Um, and both of them have the problem that they don't necessarily include the full diversity of, or I think they've been updated since, but at least a few years ago, they didn't include the full diversity of human populations, which medically is relevant because this does affect treatment reaction, and this does affect treatment response and adverse drug reactions when it comes to prescribing medications, and immune responses when it comes to transplantations, and knowing rare variants that are not so rare in specific populations, and all these sort of medically relevant um, cases. So yeah, so some nomenclature. So a linear reference means that you're basically using one DNA path for each chromosome. So one sort of standard sequence that you align with. And even if that reference is really high quality, it can still be missing DNA segments that are present in populations all over the world, but absent in the reference genome. It sometimes misses complex regions, so where people differ structurally, those structural variants, those inversions, those are quite difficult to keep track of when you're looking at a single linear genome reference, and I'll digress on that a little bit later, probably. And yeah, and so the other thing is that highly repetitive areas uh, where short sequencing, yeah, so highly repetitive areas, or these sort of tandem repeats that I spoke about. When you talk about, and one method is called short sequencing, it actually quite struggles with reading these. Um, and the yeah, well, let me actually tell you Why that is. So there's two different types or the two major different types of sequencing. You have the short read sequencing and you have the long read sequencing. So, and I'll talk a little bit about Novo assembly and all sorts of stuff like that as well. But so short read sequencing um, is essentially producing, as the name implies, DNA reads that are typically short. Short in this context is between 100 and 200 or 100 and 150 base pairs, so letters, and they're very accurate. But when it comes to these repeated regions or rearranged regions like the inversions and insertions and all of those ones, then those short pieces can't be placed uniquely. So it's like trying to reconstruct a book. If you use the analogy of continue with the metaphor of using letters and books, and there are the same sentence appears multiple times throughout the book. You won't know where you are in the book from the sentence, right? It's going to be very difficult to map where does the sentence go in the book because you can't figure it out from context. And that's the problem essentially with short read sequencing as well. An example, by the way, of a technology would be the Illumina platform. It's a type of short read sequencing platform, very common type, really, really cool as well. To use the scientific term cool, it's very interesting the way that it works. But yeah, so that's short read sequencing. And then the solution to this, in a way, is something called long read sequencing, which is a company that makes that as one company that makes that as PacBio. And yeah, long read sequencing produces thousands or even tens of thousands of bases, or reads that are bases, tens of thousands of bases long. And that's like getting entire, if you think, continue with this book analogy, it's like getting an entire paragraph. So that's easier to place in this book. The genome, in this case, in this metaphor, is the book. And yeah, and that span can repeat, or that the long read can span across repeat regions. It can span across structural inversions and breakpoints, and um, yeah, long reads essentially make it easier to see these structural variants and repeat structures. Now, specifically, the Nature article, the one that looked at the 1K-CP, the the Chinese 1,000 pangenome project. So they looked at over 1,300 participants, so people, and they produced for almost all of them, I think 1,100 and something, 116, they produced diploid genome assemblies. So what is a diploid genome assembly? Diploid, as most of you know, I just often I do just say the basic definitions because the idea behind these webinars is that anyone, whether they're medical clinician, as most of you are, whether they're a scientist or whether they're just someone interested in the specific topic, can kind of just jump in. So a diploid means that you have two copies from each of each chromosome. So that would be run from each parent. So that a diploid assembly aims to reconstruct both paternal and maternal copies rather than just collapsing them into one sort of single average. Um, the flip side of that, so if you're not diploid, you would be haploid. So that would be one copy of chromosomes. Because if you think of human chromosomes, they come in pairs. You have the one set, or you have the one half. How do I phrase it? You have half of them that you got from the one parent, and then half of them from the same ones that you got from the other parent, and they pair up. And then you have that's the technical term for it, is bivalent um, pairs of chromosome. So yeah, so genome assembly is the process of reconstructing the genome sequence from these sequencing reads, and it's like I've said a couple of times, it's like assembling an entire book from sections. So when you're doing short sections, that would be the short read sequences, or if it's long read sequences, you have the whole paragraphs, and you can kind of put it together. Now, if you already have a reference, that's great because then you can see, oh, this goes there, and I can see this goes there because even though this part is blank, I can see the beginning of the sentence and the end of the sentence is the same as the areas before and after the blank session, and I can use the reference to go there it goes. But if you don't have a reference, if you're still making the reference, that's called de novo assembly. So it just means that you're assembling without relying heavily on existing reference. Now, you can still reference other data, other types of information, which I'll talk about in a second. But yeah, you're not using a genome as a reference. And yeah, you're building the genome from scratch essentially, using only the reads that you have. Now, what you can do, and there's a so the same sort of concept where you kind of create these overhangs where when you sequence a read, the place where on the edges of it, that'll be the same as another sequence. So you kind of have these overlapping regions, and because they overlap, you like puzzle pieces, you're able to read, oh, this one was just before that one, etc. So then you can actually sequence it like that and kind of figure out where in the book it goes like that. So different methods of trying to put these things together, but put the puzzle together. The puzzle is the book, is the genome. So, yeah, and so in this example paper that I'm referring to, where they looked at the thousand pangenomes, they used 55 de novo assemblies, and over 1,000 of them were pangenome informed assemblies, meaning they used population information, and a pangenome framework. So to help assemble these genomes, even when the sequencing depth was lower. So clinically, um, why does it matter that they looked at diploid? Why does it matter that they looked at both sides? Because many important questions are about which variants it's on which chromosome copy. Um, it's important what if it which chromosome copy it comes from. If it's because it's yeah, so a lot of it is not just from not from the average, not from the one sort of readable, almost want to say two-dimensional sequence, but specifically from, let's just use an example from the paternal lineage. Yeah, that's called phrasing. Sorry, phasing. Phrasing. My analogy of the books and the words. So phrasing would be the in English, but phasing is the and genomics. If it's important which specific variant or which specific chromosome copy the variant is on. I'll elaborate on that in a second as well. So just let me continue speaking about the example first. So the pangenome that they looked at in this specific study, where they looked at the thousand pangenome Chinese project, so they had over 400 million base pairs. Um, uh, that were not present in the reference genomes. Either, I think they looked at both the ones that I mentioned earlier, the GRCH38 and the CHM13. So over 400 million base pairs that were not in those reference genome sequences. So a base pair, by the way, is the letter pair. So if you had biology in school, you'd know that a binds to a T and the C binds to a G. These letters. So the molecules, those are the pairing molecules. So if you're talking about a base pair, you're talking about that sort of binding on the DNA, the A and the T, or the C and the G. And if it's over 400 million, then you're talking about hundreds of millions of DNA letters that that were missing from the standard reference genomes. So in that study as well, they looked uh, at these missing ones, and what they found inside of these many missing ones, they were something like 26 million, more than 26 million base pairs that were in very relevant regions. So regions that included genic elements. So these are gene-related sequences, or so when I say gene-related, so that would a gene would be the what we spoke about earlier, the central dogma, where you have the gene. This is the DNA that codes the RNA that gets made into the protein. So that's the genic elements. Would refer to anything that's sort of involved in that process where there's actual genes involved. Yeah, and so there were areas that were missing from the reference genome that where they found variants in this pangenome map that they created. Um, they yeah, and so they uh, and no, okay, so I think it's important to mention. So it wasn't actually just. It doesn't specifically have to be in the genes. It can also be in DNA before or after that controls when and where genes turn on and off. The technical term for that is promoters and enhancers. So yeah, so there you can also have variants in it, say a promoter region or an enhancer region, and this will affect later on when and where the gene turns off. So the variant doesn't specifically have to be in the gene itself, but can still affect the gene even though it's not in the gene. I think that's an important point to make. So the clinical impact is that in this case, missing from the reference doesn't mean that it's non-functional. It can include medically relevant biology, um, even though it's not in a genic region, even though it's in a promoter region or an enhancer region. And yeah, so that's why, and that's a lot. Over 26 million variants that were found in sequences that included gene elements, or at least in regulatory elements, um, like promoters and enhancers. So how do you even look at all of this? How do you visualize all of this? So they use something called a pangenome graph, which is just one of the ways, or I think the only way perhaps, to represent multiple genome sequences together instead of it being one straight line. It's kind of like a map. Um, and if you look at this map, essentially you have different nodes. So these are the sequence chunks. You have the edges, which are allowed connections, and then different people's genomes sort of trace different routes through this map, right? So that's the analogy of a map. So, and where you have these different branches, these different routes that I spoke about, this allows you to, when there's a branch, you know that, oh, here's an insertion or a deletion or a different structural variant, or maybe there's an alternative haplotype. So remember diplotypes and haplotypes. So yeah, so different single chromosome difference or variable repeats, so repeat regions. So without forcing everything into this sort of average linear reference. Now, the example paper also looked at small variants, so they still looked at SNPs, um, and other small uh, indels. Indels is the shorthand for insertions and deletions, and it's exactly that. It's an insertion or a deletion. So it's either adding or removing a few letters, a few base pairs. Yeah, the paper, I think they found over 35 million small variants that were once again not included in the standard reference genomes that they found specifically in their pangenome map. They found over 110,000 structural variants. They found almost 500,000 tandem repeats, and they found something like 860,000 nested variants. A nested variant, let me just focus on that for a second. So a nested variant sounds scary. so so essentially, if you think about a structural variant, so you have a insertion, which is that paragraph. So you can think logically, if you have whole paragraph, then it's not unlikely that in that paragraph there's typos as well. So there can be snips or other variants or other repeats or any of the many variants inside of the paragraph as well. So inside of an insertion, there can also be smaller variants. That is a nested variant. So that's variants inside of the other variant. And a pangenome graph allows you to see this. So you'll see a branching when there is this variant inside of the variant, whereas the sort of standard variant coding often just treats an insertion as that, just the whole insertion is kind of the deviation. It's just one blob, and it ignores the internal diversity. But a pangenome graph actually lets you see inside of it, and actually lets you see, like I just said, the variants inside of the variant. So a tandem repeat, the one that I spoke about, the DNA pattern with the short sequences of which Huntington's disease is an example, is important for two reasons essentially. The one, or the one concept that is important would be the length, the repeat length. So how many times does it repeat? And the other one is the motif, the repeat motif. So it's what is being repeated. Is it CAG or CAC, GTC, or whatever the codon is, or the letter that repeats. So that's the repeat length and the motif. And a lot of times, clinically, this is important, the repeat expansions are known disease mechanisms. So in other words, I'll take the example of Huntington's. We did a whole webinar on Huntington's as a case study. So do go check that one out if you are interested. But yeah, so if in that specific case, the longer the repeat becomes, the more severe and earlier the onset of symptoms usually is. So is correlated to. Yeah, so it matters because all the assays sometimes only focused on the sizing, but it's not just the sizing; it's also actually specifically what the motif is. So, and the reason for that, I don't know how I'm going to explain this non-visually. So, if you think about genes being read three letters at a time, and you think about a whole sequence, you can there'll always be three letters to read, regardless of where you start. Okay, which means that if you have an inversion and the letters upside down now make a codon that tells an enzyme, oh, this is the start of a gene right here, then what essentially happens is it can start reading from a random place inside of another gene. So I'm going to try and make an example. So let's say you have one sequence. It's ATCTC, ATCTC. Okay. Um, and that's how it starts with the A, TCTC, and it encodes for one amino acid, sorry, one protein. That wouldn't actually code for a protein, but let's say it does. And now you have a little inversion there, which causes it to start reading from well, let's say the T and the C inverted. So this is not necessarily, so usually a start codon would be three letters. Um, but yeah, so let's say there's an inversion on the second letter there, then it creates a start codon, and now it starts reading from the second spot, which means the next three letters that it reads makes a completely different amino acid because you've started reading one letter on. It would essentially be like if you took an entire page of words or a written page and you moved the space bar one letter earlier for all of the words. So instead of having the brown fox jumps over the other brown fox or whatever you would have th ebro fox. So yeah, okay, so you get what I'm trying to say is the depending on where you start reading, it completely changes the wording. Um, and yeah, so that's essentially what happens when you do these structural variants, and you can have entirely different genes being coded inside of other genes using the same letters just because now there's a start codon or a stop codon being created. And that is why one of the reasons why a motif is important, not just the length of it. And there are actually other reasons as well, but I think that one is quite cool. And it also just goes to show why DNA as an information storing mechanism is so complex because it's not just a one linear read. As you can read, it can be the exact same sequence, but just because you started in a different place, you're reading an entirely different story. So that's just one of the difficulties of reading DNA and how amazing the whole polymerase or the whole transcription of enzymes into different proteins can be, because biology also does this on purpose. Well, I say on purpose, but this happens in biology as well, where functional survival based genes are sort of double coded over each other like this. Obviously, this is through billions of years of evolution, but purposeful with open quotes. So it's survival based, but it happens in nature. Let me phrase it like that. Right. So I spoke a lot about the sort of enhancer regions and the promoter regions. So there's a term called exon, which is just the part of the gene that gets transcribed all the way to the final RNA after splicing. So and contributes to the protein sequence. And this is the part that gets read in those. I've been speaking about the codons and the three letters. So that would be the exon. That's the actual region of the DNA that goes onto the different types of RNA and eventually dictates what is the protein sequence. And if a structural variant deletes or duplicates or disrupts an exon, it, like I just described, completely changes what gene gets produced. Because a three-letter codon dictates what amino acid is being made, and when the amino acid chain stops, and in that case, how the polypeptide chain forms, and ultimately what protein you get or perhaps don't get if you do sort of make that it's a non-viable protein. So, and in this example paper that I keep referring to, they identified, I think, thousands of structural variants overlapping exons. So overlapping these protein coding genes. Um, and many of these protein coding genes were in medically relevant gene sets, like cancer catalogs, for instance. So the clinical point is that structural variants are not rare curiosities, and each person can carry many, many gene altering structural variants and rare ones that would be the kind that we actually worry about the most in diagnostic works. But these can be missed in reference genomes, and that's why pangenomic maps like this is extremely important, and why pangenomic maps in very diverse populations are extremely important, so that you can try and identify these specific translocations and deletions and insertions and those type of things. So all of this refers to the mappability of it. So whether a sequence read can be placed uniquely on the genome. In highly repetitive DNA, short reads, you say they map ambiguously. So it's like having a sentence that appears 200 times in a book. Like I used the same example a little bit earlier. You don't know where in the book that goes. It's the same sentence. And clinically, that matters because a variant in a low mappability region can be systematically undercalled, and disease associations can actually be missed or mislocalized. So, and the once again, the 1,000 pangenome project paper, specifically, they specifically actually mentioned a couple of important haplotypes or structural variants that relate to traits that sit in these regions that are historically hard to read in short read GWAS pipelines. GWAS is genome wide association study. We also did a webinar on that. It was actually one of the first ones that we did. But yeah, genome wide association study would typically be the type of study where you look at all of these genomes. So genome wide association study, when you're looking at genomes, but in those cases, in those studies, it would, when you're using a reference genome or using pangenomes, and which genomes are you looking at to identify? Let's say you're looking at a drug or a disease. What are you looking at? Because you might miss it for all of the reasons that we've discussed tonight. So I can give an example, which is the HLA axis, or the HLA genes. Sorry. So the HLA is the human leukocyte antigen, and they sit in the major histocompatibility complex region, the MHC region. And they are very important for immune recognition, which in clinical settings and medical settings and surgical settings, which are all the same setting, but I like repeating things. Um, this influences things like transplant compatibility or autoimmune risk or drug hypersensitivity in some cases. And so, yeah, HLA typing resolution is often described in fields essentially. So the more fields there are, the more precise allele definition. So in other words, the more detail in the allele naming and including codons and non-codons, or sorry, coding regions and non-coding regions and exons, um, and the differences and being able to see insertions and deletions and that type of thing. And short read methods have capped resolution for the reasons we've discussed. They because they're just 100 and 250 base pairs, they often miss a lot of these things, and so that's why something called four field resolution is a lot better for capturing non-coding diversity. So, in other words, the better population specific HLA references can improve matching association studies and potentially interpretation of immune related risk. So that's one example of a clinical example of a gene family that, if we look at a higher resolution of it, a better way of sequencing this, we can directly better the way that we look at how will this affect transplantation or immune reactions. So yeah, that's why I spend so much time on the HLA as one example. It's the same example also used in the the study example that we've been speaking about. So you can read about it there as well. I will link it. Now, a term that comes up a lot in these sort of genome wide association studies or genome sequencing is QTL, or EQTL sometimes, and it's EQTL stands for expression quantitative trait loci locus. It's the one is just the plural of the other, but the what that means essentially is that a genetic variant that statistically associates with how much RNA a gene makes, so its expression levels. That's the EQTL. So if you can measure gene expression across many people and correlate it with a specific genotype, so the specific genes found in an individual, you can find variants that influence expression. Um, and the so if you take the sort of bigger version of this, the more impressive version, you get pan-variant EQTL mapping. So that means that they don't just look at SNPs, they include structural variants and transcript tandem repeats and nested variants and all of these many different variants, and they are actually able to look at the expression, how all of these affect the expression in all of these different locations where they found. And specifically, thousand, not the Thousand Genomes Project. That's a different genome project. The Thousand Pangenome Chinese project that is in the study that I'll link. They identified over 3,000, I think 3,000, almost 3,300 or 3,000, between 3,200 and 3,300 EQTLs that are involved in complex variants and that show some structural variant regions that contain many sort of regulatory signals. So exactly the type of complexity that would have been flattened in a single reference genome approach. So yeah, you can still so that so you can still try and infer a lot of data if you have less information. As a method, a statistical method called genotype imputation. So that's if you have partial genetic data, like you only have the SNP array, then you can infer missing variants by comparing it to a reference panel of known haplotypes. Like I said earlier, or a haplotype has multiple definitions, but specifically the definition that we're talking about now is a haplotype is a set of variants that are inherited together on one chromosome copy. So if we know the common haplotype patterns in a population, we can predict the missing markers more accurately. So that would be an example of genotype imputation. So you have a certain amount of gene information and you have the information about the haplotype, and therefore you can use imputation to see, oh, this is what I'm missing. But in the example paper, they look at pan-variant imputation reference panels. So that includes multiple variant types. So it's not just SNPs. So essentially what this means is that you can do better association studies and ancestry relevant polygenic risk scores and population frequency estimations, and so you can guess better. Essentially, if you have this data, you can guess better is essentially what it comes down to. So yeah, I mean, which is statistically, this is a controversial opinion, but essentially probability and statistics is just it's just better and better methods of guessing, isn't it? But yeah, mathematically backed guessing, I suppose. Mathematically and empirically backed guessing, but it's a different topic. So clinical takeaways from today's topic, this topic, the pangenome example that I used, is that you have a bias in a lot of the genomes. One of the biases specifically that I named was the Eurocentric bias, but there are other types of biases as well when it comes to these genome assemblies. That's why we have to look at many different diverse populations. That's why we have to look at many different types of variants, structural variants, right, tandem repeats, not just SNPs. Maybe I didn't mention this tonight, but there's another type of variant called a microsatellite, which is essentially just a tandem repeat. So it can also be very informative if we look at these ones. Um, and yeah, you have these population specific resources. So I use the example of allele frequency being different in different populations, and how this what could be the major major variant in one population could be a minor variant, or the minor allele frequency in a different population, which means if you, I'll give a clinical example, if you prescribe a drug that's safe for 99% of people based on a major allele frequency of a certain population, but that is the minor allele frequency for a different population. And it just so happens that that allele is also the one that contains the structural variants and variants that affect the metabolism of the drug. Then you've prescribed poorly. Then you've prescribed the drug that might not work so well for those people, or even worse, cause side effects, adverse drug effects, that type of thing. Yeah, so that's just one example. So pangenomes are a step towards more accurate precision medicine. The veil has been dropped. We've been speaking about precision medicine all night long again. It really is a big, big thing in medicine. And yeah, so if you remember nothing else from tonight, just remember that a single reference genome is not enough for equitable, accurate genomic information across populations. And for many clinical, there are many clinical relevant variants that are left out of the standard sort of genome approach: structural variants, short random repeats, microsatellites, etc. All sorts of variants that are not seen in the sort of linear approach. We spoke about indels or insertions and deletions. We spoke about nested variants. So yeah, these are all systemically undercaptured in a lot of the classical workflows. And this is essentially the problem that large population or pangenome projects try to address by improving the allele frequency estimations, the guessing, which is the central point of variant interpretation, the correlation studies, the association studies. It's better guessing, better statistics, and better sample data. And yeah, so the future is moving forward quite fast with a lot of the precision medicine really developing into some very interesting things that we can use. But in order for us to really fully utilize precision medicine, we have to make sure that the genomes the data that we're basing all of this off of, is good data, is high quality data, and high quality not just in literal DNA sequencing, but also in looking at the diversity, whether that's population diversity or whether that's looking at molecular diversity. And yeah, that's why we look at, or that's why people advocate for looking at pan-genome aware pipelines, and especially as long read costs fall. Just yeah. So that's it. That's it. All I have for tonight. So I see. Let me just double check on all the platforms. I don't see any questions tonight, but a question that I get, or that I got last time from when we spoke about precision medicine, was how do I order it? Where do I order it? How much is a kit? So it's not commercially available everywhere. You can. There are contract labs that do this type of thing in South Africa, for example, and long read sequencing run. So that would be looking at, if you use the standard PacBio system, you'd be looking at, I think, 64 samples at a time. And that to run it operationally costs about 11,000 Rand. And there should be a couple of labs that do that type of thing. I think they're in Cape Town, the CPGR probably have a long read sequencing. Yeah, they probably have a long read sequencing. They definitely have an Illumina. So short read sequencing, lots of labs and universities have access to those. So, but it doesn't tend to be a commercially available service that people offer. So you'd have to, if you want to do a long read thing, you'd have to physically contact that lab and ask them specifically, can you do this for my patient so that I can better prescribe medication to them based on all of this data. And they'd essentially have to do a specific study for you. But more and more, and as the future comes along, people are implementing these things, and it is becoming a commercial product or a service that people offer. We're already seeing precision medicine kits on a smaller scale where people do gene assays and that type of thing. So it's only a matter of time before we have genome sequencing and exome sequencing and pangenome informed gene imputation based on population ancestry etc., etc. So the scale of the precision medicine and what is included and which variants are looked at keeps increasing. And so, yeah, it's important to keep up to date with what the genetics is doing to ultimately um, better the patient's lives and better give them the treatment that really helps them and yeah, cater the treatment to them specifically. But with that, that's all that I have for you tonight. So that was all about pangenomes, and like I said, I will link the article that I kept referencing as an example too, but that's just one of many examples of a pangenome or even a multi-genome um, incorporation like this. And yeah, so you can look at a bunch of them. There's the Thousands Africa Genome Project. It's not pangenomes, but it's looking at African populations, and there's all sorts of different genome inclusion projects. But with that, I hope that sparked your interest in it a little bit in the medical population genetics, and that you would go do further reading after that. And with that, I bid you adieu. Thank you. Until next time.